And welcome back to week 12. We are now up to the meiosis and sexual reproduction part of this week's material. So we all know a big part about being alive is reproduction. It's one of the key attributes of living things. Variation is what we can clearly see in this picture is the differences between individuals. We have hereditary, which is the study of how different traits are passed on from one generation to the next, and we have genetics, which is the study of hereditary. So a moment ago, I said parents pass on traits to their offspring. They don't actually pass on traits, they pass on genes. They pass on DNA with a particular sequence. We all have two sets of chromosomes, one that we received from one parent, so we have a paternal chromosome, and another chromosome from our other parent, a maternal chromosome. And we have these stretches in the genome that are genes that are going to express different types of proteins. So we have a gene for hair color up here. We can see same location on both chromosomes from the paternal and the maternal. And if they have actually different sequences in that gene, they can have different phenotypes, or the trait will be different. And we have different genes with different variations with different alleles based on, again, what sequence is expressed. For here, we can sort of describe these alleles by having the A allele and the B allele as just being these different sequences within the genome. And again, they can either be different between our parents or they can be the same. So in this case, big C, big C, big E, or they can be different, little d, big D. We'll talk a lot more about that in future weeks. So we've already been introduced to this a little bit, but a karyotype is simply the ordered display of all the chromosomes. And technically in here, these are actually showing replicated chromosomes. They're kind of smushed together, but most of the time, again, our DNA is not undergone S phase of interphase and is hanging out in that G1. But what you can nicely see here in this karyotype are a few different types of chromosomes. You can see that here, this individual we would classify as male with an X and a Y because these are the sex chromosomes. So X and Y we're going to talk about at the end. Now, determining if it's male or female, we'll see biologically that there's a lot of gray area in depending on what definition you're using, but we'll talk about those sex chromosomes and different variations at the end. The other types of chromosomes you see here are the autosomes. Now these you receive one from one parent and one from another parent, and there's 1 to 22 of these. And those different chromosomes from the different parents, those are called the homologous chromosomes. So in week 10, we spent a lot of time going over asexual reproduction process of mitosis, copying a genome and making an identical cell. And some organisms reproduce like this. It's incredibly convenient because you only need one parent, and each offspring is a genetic clone. We're going to see this as in contrast to sexual reproduction. So we're going to see that that's going to involve two parents. And right there lies the thick of it. That the difference between these are one is an exact copy, which is very easy. You don't need to find a mate. You can reproduce with just yourself, but then everything's the same. Whereas sexual reproduction has the incredible benefit of variation, of that differences. So it's same and different. But that different comes at a cost of you need two parents. So why is sexual reproduction the more common form of reproduction in animals? Because we see evolution works off of diversity. Having variation, we can see, is the key to survival through evolution and natural selection. You can ask yourself, quiz yourself, of course, mitosis is asexual, meiosis is sexual reproduction. And now we're going to have to start getting into the nitty-gritty of terminologies and to actually think a little bit about life cycles. We are going to be focusing on the life cycle of humans, where we're going to see that only very specific cells undergo meiosis, undergo this very important process that only happens in our gametes. Our somatic cells, as we learned earlier, they're undergoing mitosis. So we're going to see that in these cells undergo meiosis, and they're going to end up having the amount of DNA within their genomes. So we have diploid organisms, which we are, and there's particular cells that are undergoing mitosis and going to have their DNA to become haploid. Those two halves, that egg and that sperm, which we'll talk about in a second, are going to merge and going to form a zygote in fertilization. And then this is what we covered earlier, that mitosis and that development of that cell reproducing. Other organisms, particularly fungi and plants, have a different route where the main thing that, like the living part that you think of, isn't actually the diploid stage, but is rather the haploid stage. So where is meiosis occurring? So here we have the anatomy for a male, and hopefully based on that previous slide, you can have identified it's occurring in the testes. This is where the gametes are. Everything else has two copies of the genome and is diploid, but the testes is where this meiosis is going to occur, and that two copies are going to be converted into one copy. Now, as we can see here, we're about to go through this process of meiosis, but we're going to see for men producing the sperm, it's pretty much exactly what we're going to see, where there is a first round of meiosis and then there is a second round of meiosis. So one cell is going to produce 4 daughter cells. And that's what you see in sperm development. Whereas in females, equally have all of their somatic cells expressing 2 copies of their genome as diploid organisms. But in their ovaries, where producing their eggs, is where meiosis is occurring and that DNA gets cut in half. Now, the fascinating and interesting thing and difference between meiosis in females and males is where in males you get 4 daughter offspring from one cell. In women, in oogenesis, you actually have one cell taking the majority of the cytoplasm. So we're going to see there's 2 rounds of meiosis, and that first division, which is what happens before birth for females, and this cell gets stuck in this after this first round in prophase 1, but the splitting doesn't occur evenly. And anybody who knows anyone who's undergone fertility treatments might be familiar with the maturation of follicles and releasing the oocyte, which is actually in that first round of meiosis. But once again, when that cell goes under that second division, instead of evenly being split to produce 4 different daughter cells that are equivalent, it's an uneven splitting again to really only produce 1 egg. 1 haploid ovum. Okay, so here for the next few slides, I go over some of the same things I've been saying about that you have autosomes, which are chromosomes 1 through 22, and you have the sex chromosomes, where we have 2 copies of each chromosome, one that we got from our mother and one that we got from our father, which is why we are a diploid organism. We have diploid cells that 2 copies. So now we're going to get into the nitty gritty for meiosis. But before we do, we have to remember that interphase is the same for all of these cells, meaning that those cells are first undergoing S phase and that duplication of the chromosome to make 2 sister chromatids. So again, we have G1, where those chromosomes are just those double helixes, and then it undergoes replication. Each chromosome for the maternal and the paternal have duplicated. So you can see here how each chromosome after S phase and after replication is made up of those 2 sister chromatids that are attached at the centromere. Just to identify some of these different things again, you have different chromosomes. Here we have an example of chromosome, we'll call it 1, 2, and 3, where you got 1 chromosome set from your maternal, 1 paternal set. And within those pairs, those homologous pairs for chromosome 1 from your maternal and 1 paternal, chromosome 2 homologous pairs, and this would be homologous pair 3, each made up of, again, 2 sister chromatids. So we are now going to talk about meiosis and how these gametes, these very specific cells, cut their DNA in half. We're starting from after DNA replication. Of course, we could all think about ways that we could design this differently, but that is not what has happened in evolution, so this is the process of how it occurs, where we see these 2 duplicated chromosomes are going to ultimately get split and result in 4 daughter cells. So the good news is for this process is you're familiar with the terms. You're already familiar with interphase, like we said, G1, S, and G2, and you're actually familiar with the terms for these phases of meiosis, which are prophase, metaphase, anaphase, and telophase. And we're actually going to see that what makes up meiosis is just 2 rounds, essentially, of mitosis in terms of what the chromosomes are doing. So we are going to see that an interphase is when chromosomes get duplicated in the S phase. And we're going to see now that there are 2 rounds of meiosis, where in the first round of meiosis, the homologous chromosomes separate. And in the second round of meiosis, it's the sister chromatids that get ripped apart. So it's this first round that is significantly different than what we saw in mitosis. Okay, so this is a great slide to check your understanding and to see if you can fill out what are the names for the different parts. What are we describing here? What is the term for the difference between these chromosomes versus these 2 parts? What is separated in meiosis 1 and what is separated in meiosis 2? As always, fantastic video as well to check your understanding. Okay, so hello, I turned on my camera because this part, there's going to be some things that just being able to show you my hands is going to be helpful. So what I'm going to go through now is going to be what's on all the next slides. So I'm going to just do it in this one slide, and there's going to be a bunch more pictures. We're showing you the same thing because, again, a lot of these things you want to just be able to identify what's going on in the pictures to be able to say what's happening to the chromosomes, what's happening to the microtubules, where is the nuclear envelope, and to be able to describe these. You are going to be making a video where you are going to be physically moving the chromosomes to show me that you understand this process of meiosis. What we have in meiosis are 2 rounds of prophase, metaphase, anaphase, and telophase. And what we learned about in mitosis in terms of what was happening to the chromosomes, microtubules, and nuclear membrane in mitosis, it's almost the same thing that we're going to see here in meiosis. So first, you have interphase. That's what's happening before meiosis starts. Again, G2 phase. The cells have already duplicated their chromosomes, so they're in these X shapes that we're familiar with. So the first stage is prophase 1. Now, prophase 1, one of the most important parts of meiosis is occurring, where we're going to see the homologous pairs of chromosomes are lining up together. So you're going to have maternal 1 chromosome line up with paternal chromosome 1. Maternal chromosome 7 is going to line up with paternal chromosome 7. And there's actually going to be an event called crossover, which I'll explain at the end of this. So there's some extra events happening in prophase 1, but the same things that we saw previously in prophase, meaning that nuclear envelope falls apart. Those microtubules, those are growing and forming and starting to attach to those chromosomes. The chromosomes themselves are going from being open to condensing into that small bit that can move around. So prophase, the same things you learned about in mitosis are going to happen in meiosis. It's just that now those homologous pairs are in a complex. So when we get to metaphase, again, the second stage, in this case it's metaphase 1, all those homologous pairs are going to have lined up in the middle. And what we see in anaphase 1, it's those homologous pairs of chromosomes that are going to get ripped apart. Now remember, each single homologous pair is actually still made up of those 2 sister chromatids. So in anaphase 1, it's the homologous pairs of chromosomes that are separating. In telophase 1, the same things that happen in telophase in mitosis are occurring. And here's where we can all state, yeah, if we were going to be designing the system, it's not that efficient. We're going to see everything that happens in telophase 1 is going to happen in reverse in prophase 2. It doesn't matter about what logically makes sense. This is how it worked out in evolution, which is that you work with the system you got. Just like we could think of, it's not great to have the same hole that we eat out of and that we breathe out of, but that's evolution for you. So back to meiosis. After anaphase 1, you have telophase 1, where those chromosomes are going to open back up, that nuclear envelope is going to form, and those microtubules are going to get shorter from the ones that were attached to the chromosomes, and there's a few that are elongating to stretch out that cell because it now needs to still undergo cytokinesis, the separation of the cytoplasm. So we can see here, we've got diploid cell. That cell started diploid with 2 copies of each chromosome, one maternal, one paternal. At anaphase, those 2 homologous pairs of chromosomes separated, so that by telophase 1, we now have haploid cells with only 1 copy of each chromosome. Whether or not it's from the maternal or paternal side is going to depend, and we'll talk about that in a moment. So after this first round, meiosis 1, we now have the homologous pairs of chromosomes separated. We then go right into meiosis 2. There is no interphase where S phase and DNA replication occurs again. So there's no break in between and no DNA replication. Instead, we go right into prophase 2. Now, prophase 2, as we know, the nuclear envelopes that are just formed are going to come apart. Those spindle microtubules are going to form and going to start reaching out to grab the kinetochores, which is in the centromere of that chromosome. That chromosome is still made up of 2 sister chromatids. You can see here and here. So prophase 2, those 3 steps. By metaphase 2, those chromosomes are going to have lined back up in the midline plate. Now here, meiosis 2 we see is pretty much the exact same thing as mitosis, where each of those sister chromatids is attached to opposite poles and are going to be pulled in opposite directions. That pulling is going to, of course, occur in anaphase 2, so those sister chromatids are being ripped apart in anaphase 2. Then, of course, finishing it off with telophase 2. And this is where those nuclear envelopes are going to reform. Those spindle microtubules are going to fall apart. And those chromosomes are going to open back up so that they can do transcription. So all of these cells are haploid, meaning they only have 1 copy of each set of chromosomes. So I have a whole bunch of pictures now going through everything I just said. But to again go over high level, we have interphase, followed by meiosis 1, which has got 4 parts: prophase, metaphase, anaphase, and telophase, with most interesting things happening in prophase 1, which I'll talk more about now. And then that goes right into metaphase 2, which is prophase 2, metaphase 2, anaphase 2, and telophase 2. So again, meiosis 1, it's the homologous pairs of chromosomes. So maternal chromosome 7, paternal chromosome 7, they are going to line up and form a tetrad, form a complex, and then it's that complex that is going to get ripped apart. So homologous maternal chromosome and paternal chromosome get separated. In meiosis 2 is like mitosis, and it's the sister chromatids that are ripped apart. So this says the same thing, everything we just went through. Where at the end of telophase 2, there is that second round of cytokinesis, where that's the physical cell membrane separating out and forming those now 4 daughter cells. So you want to be able to recognize what is happening in each stage, either by words or by pictures. So can you explain this? And again, you will see this because you'll be explaining it in the video you're making to turn in for your assignment. So now we're going to talk a little bit more about some of the variation and what is making meiosis be far more favorable for most animals. So it all again has to do with genetic diversity. So genetic diversity is caused by those changes in the nucleotide sequence. And when there's a sequence that's different between 2 different genes, that's called an allele. Again, we're going to talk more about this vocabulary in the unit 4. We're going to see there's 3 main things that account for this variation, for this genetic diversity that sexual reproduction leads to. So this is why sexual reproduction has variation for these 3 different mechanisms. 1 is the independent assortment of chromosomes. 2 is this crossover event that I mentioned but didn't really get into. Then there's random fertilization. So What did I mean by independent assortment? How those chromosomes line up is independent of other chromosomes. So you've got chromosome 1, maternal and paternal. Those are going to form a tetrad. But how that tetrad forms relative to chromosome 2 and 3 and 4 and 5 and 6 and all the way to chromosome 23 or the sex chromosomes is all based on random orientation. So in one case, you can have the maternals all lining up on the same side, or you can have opposites. And given that they're going to get ripped apart in that first round of meiosis, in meiosis 1, those homologous pairs get separated. Whereas in metaphase 2, you can see the different orientation is going to lead to different cells. So how those chromosomes line up can lead to different combinations. We, as humans, have 23 different pairs of chromosomes. So even without the crossover event I'm about to talk about, there are still so many different options so that the daughter cells, so that the cells that were formed are different always than the parent cell. Okay, so the second root or mechanism for diversity, for this genetic variation, for how children look like a combination of both their parents, is when I say there's a maternal and a paternal chromosome and they line up. During prophase 1, they actually do another process called crossover. And this is where they change up their different segments of DNA and they swap them so that each chromosome is a little bit different than when it started. So that there isn't then one paternal chromosome and one maternal chromosome, but each one are sort of a combo. It's that shuffling of the deck. So we can see here physically how it happens. It's complicated. And this is actually why sort of species are different species, because these chromosomes have to line up perfectly so that when they swap parts, they're swapping the exact same part of the chromosome. So it's the same genes that are being swapped. And it's called a synaptic complex. And there's different proteins that are sticking them together, and then they actually form, and then you can see here, they form this crossover where the two different chromosomes literally cross over. And so this is where you're going to swap parts. So it's like swapping arms for an arm. So this is where in prophase 1, you form the tetrad where the synapsis is occurring, and the crossover event where you're swapping parts is going to occur so that in anaphase 1, each of those chromosomes, each of those sister chromatids is actually a little bit different from each other. So that again, each of those daughter cells is going to be distinct from that cell in which it came from. You can again check your vocabulary by filling out this slide. You can also watch the video. And it brings us to the third mechanism for genetic diversity within sexual reproduction. And that is simply sort of summarized by this idea of there's a lot of fish in the sea, that you have half of genetics that is going to combine with another haploid cell and can form that zygote. Well, so what half is joining with what half? Now we can argue it's not totally random, but genetically it's random. It's random who those different which those different gametes are going to come together. And we can see if we look at different animals that sexual reproduction is significantly favored evolutionarily, because that's what we see is the form of reproduction by the vast majority of animals. Again, because what is surviving is going to be able to survive if there's more variations. Natural selection fundamentally works on the variations and the differences within a population. So you should now be able to compare and contrast meiosis and mitosis. I've got some different sort of summaries here and some sort of comparisons that honestly, it's not going to do you well to have me go through it orally, but rather for you to sit through it and look through these and quiz yourself. Fill out this table without looking at the next slide and seeing, can you answer these questions? And then I put in the right answers. Okay, so this all brings us to the final part of this chapter, which is all about non-disjunction. And what non-disjunction is the chromosomes during meiosis, So either during meiosis 1, being those homologous pairs of chromosomes, or meiosis 2, and those sister chromatids don't separate appropriately. And what ends up happening in non-disjunction is that you are going to get one cell that is going to have too many chromosomes, and you're going to get another cell that doesn't have the right number, has too few. Or sometimes it actually isn't the full chromosome, it's just a part. So non-disjunction is all about when those chromosomes are getting ripped apart, it not going correctly and having one go on one side where it should have been split up. So you can see how it plays a part in different roles. It can happen either in anaphase 1 where it got pulled apart inappropriately, or it can get pulled apart inappropriately in anaphase 2. Ends up being called aneuploidy. So any chromosome abnormality where it is either too few or too many is aneuploidy. And you probably are familiar with some individuals who have either a trisomy, or you may even know someone that has a monosomy. So a trisomy is when you have three copies of a chromosome. And the most common for this for humans is Down syndrome, which I'll talk about more in a second. Polyploidy. In animals, it is tightly regulated and there's not much room for variation. So polyploid organisms, it's usually deadly and lethal before that organism survives enough to be born. Whereas in plants, plants have a lot more flexibility there, and most of the plants you eat that are delicious are actually polyploid. But there are some cases in humans where it is not a lethal case, and there are individuals born. As I just mentioned, there's Down syndrome. Down syndrome comes mostly from trisomy 21. Occasionally, it actually forms not from having a full third copy of the 21st chromosome, but rather just a particular gene. And the incidence of Down syndrome are much more prevalent as the age of the mother increases. So trisomy 21 is Down syndrome, and you can see that 3 copies here. And individuals have characteristic facial phenotypes and other developmental disabilities often. So where in humans we have a lot of flexibility with chromosomes is really only our sex chromosomes. And so this is where I mentioned earlier that typically the definition of female would be XX and the definition of a male would be XY, but even in those most simplest terms, it doesn't really work out in biology. In fact, sometimes a definition of female would be to have a bar body, which is actually that inactivated X chromosome. So you have 2 copies, but you only need 1. So having a bar body, which is what that second chromosome turns into, that second X chromosome, used to be a definition of female. But we're going to see how even in that doesn't actually demonstrate what reality is for human biology, which is that there's individuals with Klinefelter syndrome, which is approximately 1 in 1,000 live births. So it's not super common, but it's also not that rare. Think about if you've ever met 1,000, what you would define as men in your life, you may have met someone that has Klinefelters. And what that means is that they had a trisomy, or in this case, actually 4 copies of the X chromosome. So typically in men, you just have one X and one Y. The X received from the maternal side, the Y received from the paternal side. But there's more range. So Klinefelter syndrome, that's with extra X chromosomes in males. So they have that Y. Whereas, I'll move this up. In Turner syndrome in females, which happens less frequently, but that's monosomy, where there's only one X chromosome. There's only one X chromosome. So in this case, if you're going by the definition of who has a bar body, Turner syndrome would be considered a male, even though they have female genitalia, whereas Klinefelter syndrome, which has, again, male genitalia, would be classified as a female because they're going to have bar bodies from their X chromosomes. Another case, there's some different terms for sometimes super Y, also about 1 in 1,000, which is where a male individual will have 2 Y chromosomes. So in that case, you know the parent which had that non-disjunction event. It was the paternal side that was donating that Y chromosome. And this is another case where previously there was research that suggested something about the characteristics of these individuals, but then upon further research, wasn't the case, which is that they were particularly aggressive and that was causing some criminality. But that actually further research showed that that was not the case. What is the case is that these are men that are typically taller, that do have some elevated levels of testosterone and have the effects of bad acne and some other effects. So that's another case where, again, how you're defining gender and sex is very fluid based on what definition you're talking about. Um, so other examples of events that lead to these crossover errors would be crit de chow. Anyone speaking French can correct me, but that's where you have an issue with the fifth chromosome, and so this is also a rare but more common than you would think event where you're missing just part of the fifth chromosome, but other parts are remaining. So if you're missing the entire fifth chromosome, that would be a lethal event. But there's are examples of cases, and it has to the title of it has to do with the sound of their voice when they're babies that sounds like a cat screeching or crying. So Fragile X will be the last disease they'll give an example of, which is actually not a case of non-disjunction, but rather where there's a mutation in a particular gene. In this case, it's the FMR1 gene, where there is a different sequence. It's a different repeat of C's and G's, and they can either be super long, which then causes that section of the chromosome to be very fragile, is what you say, because it's really long and thin. And this is a problem depending on whose context, about 1 in 4,000 or 1 in 6,000. And because it's on the X chromosome, we see that there's a difference in females and males. And we'll talk more about that next week in genetics. So what we'll also cover more in genetics, but this is also reminding us back from last week, is thinking about thinking about the 2 weeks ago, and thinking about translocations, deletions, inversions, and duplications, which are very common when crossover happens. So when crossover gets screwed up, these are some of the results. So you can quiz yourself and hopefully you can see here, this would be a duplication of this section. We can see here there is an inversion where GHI gets switched to IHG. Here we can see there is a deletion, C got removed, and then here we can see a translocation where you're actually getting that swapping, but instead of it being those homologous pairs of chromosomes, chromosome 7 and chromosome 7, in this case that would have been chromosome 8 and chromosome 15 or something. So you should now be able to answer these questions. And if you are confused, come to see a tutor, come and click on me and set up a meeting for my office hours, or email me. Be well.